this kind of free radicals to destroy some intracellular pathogen. So certain type of intracellular pathogen only works with a very deadly toxic agent like the RNS. And the other kind of killing mechanism occurring in the phagolysosome. So first we know oxygen radical. Second, we know nitrogen radicals, right? Free radicals. So third is inside the lysosome itself. I mentioned it contains originally antimicrobials compound, right? So they are known as the preformed antimicrobials, meaning they are already there originally without initiation of any uh, without initiation from any pathogens. So they don't have initiation from the infection to produce the preform. They are already there inside the lysosome. So which includes cathepsin, enzyme, lysozyme, lactoferrins, hydrolytic enzymes. So all of the enzyme has a specific function. They can damage bacterial membrane or cell walls, or they avoiding bacteria from getting a sufficient nutrients, lactoferrin, or they can hydrolyze the bacteria proteins. So these are basically antimicrobial kind of killing mechanism used by the immune response to kill the pathogens. So if we look at type of infection, some patient they might have a disturbed process of this killing mechanism. So for a healthy people, they have all of this mechanism. But some patient, it turned out that they are getting disease because they have defects in oxygen dependent killing mechanism. So because we know that oxygen radicals is very powerful, right? They can kill a very strong bacteria. But some people they lost this ability, although they could actually replace their inability by the preform. So here the preform antimicrobials product usually present in a lot of in healthy individual, in a lot of people, because they are a common protein in the cells, right? Unlike free radicals, it an active mechanism. It require initiation, so it require healthy process by the body. So if the patient is not healthy, there might be possibility that they cannot perform the oxygen dependent killing mechanism, and therefore some patient they could be getting some serious infection in general because they lost some of the killing mechanism through phagocytosis. Okay. So these are the summary in diagram form. So I'm not going to go in details. It's just a summarize the whole thing, a summary of the whole thing. And I mentioned just now, in general, phagocytosis would be working in majority of the bacteria, except that those who have a protective barrier on the surface, right? Because for the phagocytosis to be conducted, they require recognition on the surface, isn't it? Therefore, when the bacteria have surface on there, have a protective coating on the surface, it will not allow phagocytosis to occur. So there are actually a lot of other mechanisms on how the pathogen can escape phagocytosis besides having a protective mechanism on the surface. So here you can see there are a lot of mechanisms. Some of them avoid contact with the phagocytosis. Meaning how? Meaning they try to remain, they stay in a certain region of the host tissue which is inaccessible by the phagocytes. So, for example, lumen of the glands is not accessible by the phagocytes, urinary bladder, or surface tissues and broken skin, right? Because we don't have mouths, right, on the surface of the skins. And the other type of pathogen, they will avoid provoking inflammation response. So we know that inflammation is one of the innate immunity response that is initiated when there is an infection. So once inflammation is stimulated, it will signal any other type of immune cells to come over and help the process. So for that, to avoid that to happen, some pathogen, they even have the mechanism to avoid provoking inflammatory response. The other type is they inhibit phagocyte chemotaxis. So if you look at the mechanism, how the bacteria avoid phagocytosis is basically on a basic, each step by step, how the phagocytosis mechanism is conducted by the immune response. So first, because we know that for the phagocyte to come over and perform phagocytosis, they need to be signaled, right? They require signal. Therefore, some bacteria, they inhibit these signals by inhibiting phagocyte chemotaxis. So even sometimes the certain bacteria, they produce toxin, streptolysin, as ceftriaxone, which can kill the phagocytes. Or for example, M. tuberculosis, they inhibit the leukocyte migration. Or Clostridium toxin also can inhibit neutrophil chemotaxis. So the other mechanism is they avoid contact. The other mechanism of avoiding contact with phagocytes include covering their surface. So here I mentioned just now through capsule, a protective layer, so that phagocyte cannot recognize. Or they could also produce a layer that is very similar to the host cell, seen as self by the host. So that host thought that is actually host cells. Because, for example, they produce salicylic acid. So this kind of protein is basically a common protein that usually present also in the human tissues. Or the hyaluronic acid. Remember, this a very common compound you will come across in the cosmetic nowadays, right? They put hyaluronic acid for anti-aging. So these are common human tissues. Healthy tissues should have this acid. Therefore, the bacteria could actually synthesize this on the surface. Therefore, immune cells would not be able to recognize them. Okay. So, and the rest of the slides is basically here you can see, because phagocytosis process involve engulfment, right? So there will be some group of bacteria have the ability to inhibit phagocytic engulfment. So some of this pathogen, they have already recognized mechanism of host tissue. Why? Because these are the same mechanism used by our species over the time from our ancestors, because this is innate immunity, right? So that's why some microbe, they develop the ability to come over to avoid the process of innate immune system. So inhibition of phagocytic engulfment includes many different type of protein on the surface of the bacteria or pathogen. Or some of them, they don't secrete anything to protect them, but instead they actually survive inside the cells. Okay, and then all of this. So